Usually when we talk about getting a medical diagnosis, there's this underlying expectation of precision. Like, it feels kind of like engineering.
Right. Very mechanical.
Exactly. You fall, you break your arm, the x-ray shows that jagged white line on the screen, and the doctor just points at it and says, there it is, that's the problem.
Yeah, simple cause and effect.
Right. But then you look at complex autoimmune conditions, things like inflammatory bowel disease or Crohn's disease, ulcerative colitis, and suddenly that x-ray machine is just, well, it's completely useless. We've known for a long time that genetics play a huge role in your risk for these diseases. But there is a massive, incredibly frustrating catch.
Oh, absolutely. The non-coding issue.
Yes. 90% of the genetic variants linked to these conditions are hidden. They're in the non-coding regions of our DNA. So for the listener, it is exactly like having a treasure map with a giant red X covering like an entire city block, but you have absolutely no idea which house, let alone which specific room the treasure is actually sitting in.
That's a perfect way to look at it. It's a map that gives you the general neighborhood of the risk, but completely fails to give you the specific street address.
Yeah.
And historically, that lack of functional resolution has been a massive roadblock for researchers trying to actually develop targeted therapies. I mean, you can't fix a house if you don't know which one is broken.
Which is exactly why we are jumping into a monumental new project for today's deep dive. It was just published in Nature in June 2026, and it's called the IBDverse project.
The scale of it is just wild.
It really is. They took biopsies from the terminal ileum, the rectum, and blood from over 400 individuals, which included over 100 people who actually have IBD. And what they found might finally give us those exact street addresses. But before we get into what they discovered, help me understand the baseline here. How do we even start looking for specific genes if they are hiding in this dark, non-coding space of our DNA?
Well, to really understand the leap this paper makes, we have to look at the tools we usually rely on. The first tool is genome-wide association studies, or GWS.
Okay.
That's what gives us that giant X on the city block.
Yeah.
GWS finds the zip code of the disease risk by, looking for genetic variations that are just really common in people with the disease compared to healthy people.
Right, so it flags the general area.
Exactly.
Yeah.
But to find the actual house, we use something called eQTLs, which is short for expression quantitative trait loci.
Okay, let's unpack this. So the GWS says, hey, the problem is somewhere in this neighborhood. And the eQTL says the problem is specifically this dimmer switch on the living room wall.
Precisely.
Oh.
The eQTLs are the highly specific genetic variants that actually dictate how nearby genes are turned on, turned off, or dimmed.
Yeah.
But here is the massive challenge that this IBDverse project finally solves.
What's that?
Previous studies look at these eQTL light switches using bulk tissue sequencing. They basically took a piece of gut tissue, put it in a metaphorical blender, and studied the resulting smoothie.
A gut smoothie? Appetizing?
Right. The problem is, when you blend millions of epithelial cells and immune cells and stromal cells all together, you completely miss the vital, rare details. If you blend a strawberry, a banana, and a single microscopic jalapeno seed, you're just going to taste the fruit.
Because it just gets lost in the noise.
Exactly. You won't find the seed, even though it's the thing actually burning your mouth. So the IBDverse project completely bypassed the blender. They looked at 2.2 million individual cells at single-cell resolution.
I love that smoothie analogy. So basically, bulk RNA sequencing is like trying to listen to one person's specific conversation in a crowded football stadium. You just hear this indistinct roar of the crowd.
Yeah, perfect.
But single-cell sequencing is like putting a personal microphone on all 2.2 million people in that stadium so you can hear every single whisper. So here's my question. If we zoom in this closely and we put a mic on every single cell, do the actual rules of genetics change depending on how closely we listen?
What's fascinating here is that the rules absolutely do change.
Sure.
Yeah. The researchers looked at the data through 3 different lenses of resolution. They had all cells combined, which is our smoothie. Then they zoomed in to major population, like grouping all T cells together. And finally, cell type, which is hyper-specific, like looking at just one specific kind of memory T cell.
Okay, so 3 levels of zoom.
Right. And they discovered that while the bulk resolution finds the most affected genes overall, the highest resolution finds completely distinct regulatory variants that are completely masked in the bulk analysis.
Yeah, wait, completely masked?
Yes. Nearly 1/3 of the specific genetic light switches they found were totally invisible until they looked at the single-cell level.
But logically, why would that be? Like, If a genetic risk variant is causing something as severe and systemic as Crohn's disease, why would it hide so perfectly in just one specific type of cell?
It comes down to architectural distance and, interestingly, evolution.
Oh.
The study meticulously mapped exactly where these switches live in relation to the genes they control. In the bulk smoothie cells, the eQTL switches were relatively close to the gene. We're talking a median of 52 kilobases away for what we call the transcription start site.
Which is the main on and off switch.
Right, the main switch. But the cell type specific eQTLs, the hidden ones,
Right.
they were located a median of 200 kilobases away.
Wow, so way further down the DNA strand.
Exactly. They don't live in the main switch on the wall. They live in regions called enhancers, which act a lot more like remote controls.
Okay, so the bulk analysis finds the light switch on the wall, but the single cell analysis is finding the remote control shoved deep between the couch cushions.
Yes.
But I'm still caught on the evolutionary aspect you mentioned. If these gene mutations are causing debilitating chronic diseases that can, severely impact survival, shouldn't natural selection have weeded them out thousands of years ago? How are they surviving in our gene pool?
That is the exact paradox this paper addresses, and your couch cushion analogy perfectly explains it. Yeah. If you have a mutation in a promoter, that main switch on the wall, it breaks the gene in every single cell in your body. That is usually lethal or so damaging that natural selection weeds it out immediately.
Because the person doesn't survive to pass that mutation down.
Exactly. But if the mutation is in an enhancer, the remote control, it might only break the gene's function in one specific cell type, like a rare dendritic cell in your gut.
Oh, I see.
It only causes a problem under very specific conditions. Because of that, the variant can hide. It survives natural selection, gets passed down through generations, and only rears its head when that highly specific cell is activated.
That is incredible. The disease is literally hiding from evolution in our most specialized cells. Okay, I want to do a quick 5-minute recap for you listening to kind of lock this first part in. Let's do some bullet points.
Good idea.
Bullet 1. GWS gives us the genetic neighborhood of a disease, but eQTLs are the specific light switches that turn the genes on or off. Bullet 2. Studying bulk tissue is like drinking a smoothie. It completely misses the highly specific cellular misfires happening at the micro level. And bullet 3. High-resolution mapping proves that disease variants evade natural selection by hiding in distant enhancer regions those remote controls affecting just one cell type at a time.
That's a great summary.
Yeah.
And that realization opens the door to an even deeper layer of complexity. Because these cells and the genetics inside them are not operating in a vacuum.
Right. The environment around the cell has to play a role. I mean, my DNA is exactly the same whether I'm perfectly healthy or if I'm currently fighting off a terrible gut infection. But how my genes actually behave in those two scenarios is drastically different.
Which is why the researchers mapped what they call IQTLs, interaction eQTLs.
It's interaction eQTLs.
Okay. These are genetic effects that only show up, or get significantly worse, during a specific environmental state. They looked at variables like age, sex, and smoking. But the most important state they looked at was active inflammation.
Which makes sense for inflammatory bowel disease.
Exactly. Out of over 1,000 of these interaction eQTLs they found, a staggering 78% were triggered specifically by gut inflammation.
So the inflammation is essentially acting like a chemical developer in a dark room. It's washing over the cells and revealing genetic risks on the film that were completely invisible just a moment before. What did that actually look like in practice?
One of the clearest examples they found involved something called metallothioneins.
What?
Metallothioneins. In the cells lining your gut, your enterocytes, there's a biological pathway responsible for binding metal ions. When the gut gets severely inflamed, this pathway goes into absolute overdrive. Specific genes begin churning out these metallothionein proteins, and their job is to bind to toxic metal ions, neutralizing them, to protect the gut tissue from the extreme collateral damage caused by the inflammation itself.
Oh, wow. So they act almost like metal trash cans.
Metal trash cans, yeah.
They are literally sweeping up the toxic metallic waste that the inflammation is leaving behind in your gut.
I really like that mnemonic.
Mm-hmm.
Yes, they are the metal trash cans. Now, if researchers only ever studied biopsies from healthy individuals, the genes controlling these vital trash cans, and more importantly, the genetic variants that break them would never show up on the radar.
Because there's no inflammation to trigger them.
Exactly. You literally have to study the tissue while it is actively under siege to see where the defensive walls are crumbling.
Okay, here's where it gets really interesting for me. We now know how to find the switches, and we know that these switches change their behavior under the pressure of inflammation. So who is actually holding the broken switches? When the IBDverse researchers looked at the data, what specific culprits did they uncover? And how do they prove that a specific switch is actually pulling the trigger on IBD?
They prove it through a rigorous statistical method called colocalization.
Colocalization.
Right. Imagine you find a fingerprint at a crime scene. That's the GABIOS risk variant showing disease risk. Then separately, you find a fingerprint on the murder weapon, that's the eQTL, showing altered gene expression. Colocalization is the mathematical proof that both of those fingerprints belong to the exact same person.
Oh, that's a brilliant way to explain it.
Using this, the paper successfully colocalized effector genes for over half of all the known IBD risk regions. And they found 74 entirely new ones that nobody had ever linked to specific cells before.
Over half. That is a massive leap. We went from a blurry map of a city block to having the exact street addresses for over half the suspects. So who were the worst offenders? Which cells were harboring these broken remote controls?
The most frequent culprits turned out to be myeloid cells, and specifically a type of cell called dendritic cells.
Dendritic cells, if I remember my high school biology, those are basically the gut's border patrol, right?
Spot on.
They sit on the lining of the gut, grab particles floating by, and have to make a split-second decision. Like, is this a harmless piece of a strawberry, or is this a deadly salmonella bacterium?
Yes, exactly. They sample their environment and present these particles to the rest of the immune system to either call off the attack or declare war. Maintaining peace with harmless things like food or friendly gut bacteria is called oral tolerance.
Oral tolerance, got it.
And what the researchers found is that in IBD, a critical communication network inside these dendritic cells is broken. It's called the Notch signaling pathway.
Okay, I want to keep us out of the alphabet soup of genetics here because it's easy to get lost in a list of gene names. But what is this Notch pathway actually doing for the Border Patrol?
Think of the Notch pathway as the walkie-talkies the Border Patrol uses to communicate with headquarters. To make those walkie-talkies work, you need batteries. The researchers pinpointed two crucial genes that act like the batteries for this system. They're called MML2 and ZMIZ1.
Okay.
The study found a highly specific genetic variant that essentially drains the battery on MML2, lowering its expression specifically in these dendritic cells. And that exact variant colocalizes perfectly with ulcerative colitis.
Wow.
Similarly, a variant that drains the battery on ZMIZ1 colocalizes with Crohn's disease.
So if the batteries in the walkie-talkies die, the Border Patrol can't communicate. They panic. They lose that oral tolerance we talked about, and suddenly they start treating everything like a threat. They attack the friendly bacteria, the strawberry, the gut tissue itself, leading to a massive cascade of immune dysfunction.
That is the exact mechanism. It shows us that the immune system in an autoimmune disease isn't just randomly angry. It is operating on broken intelligence because of these specific cellular switches failing.
Okay, let's lock this in before we look at the other side of the equation. Time for our second 5-minute recap and bullet summary. Bullet 1. Inflammation acts like a chemical developer on a photograph, revealing hidden genetic risks called iQTLs, like our middle trash can genes.
Right.
Bullet 2, using colocalization, the IBDverse matched the crime scene fingerprint to the murder weapon, tying over half of known IBD risks to specific cells. And bullet 3. Dendritic cells are Border Patrol are major culprits. Broken Notch signaling drains their walkie-talkie batteries, ruining the gut's ability to tolerate friendly particles.
Perfect.
Which really makes you wonder, is an autoimmune disease an overactive immune system, or is it just a profoundly misinformed one?
It's an essential distinction. But the misinformation driving the immune system is really only half the story. And this perfectly sets up the next major finding of the paper. Because IBD isn't just about an immune system attacking the wall of the gut. It is also about the wall fundamentally forgetting how to rebuild itself.
Ah, the barrier breakdown. So now we're moving away from the immune cells and looking at the epithelial cells, the colonocytes and enterocytes that actually make up the physical lining of our intestines.
Correct. For these cells, the researchers focus heavily on something called the Wnt signaling pathway.
WNT.
Right.
Yes.
If Notch signaling is the Border Patrol's radio, Wnt signaling is the instruction manual for the bricklayers trying to rebuild the wall. It controls stem cell renewal and fixing the gut barrier when it gets damaged. To understand how critical this is, the paper brings up a really fascinating clinical case, Kindler syndrome.
I'm familiar with Kindler syndrome. It's an incredibly rare, devastating genetic condition. Patients with it have tissue that is profoundly fragile, like their skin, their internal linings. They blister and tear at the slightest touch or friction. And it's caused by a mutation in a single gene called FIRM-T1.
Now connect the dots. The IBDverse study found an eQTL, a dimmer switch, that reduces the expression of that exact same gene, FIRM-T1, specifically in the colonocytes of the gut.
No way.
Yes, and that drop directly colocalizes with common IBD risk. This proves that the disease isn't just about the immune system attacking healthy tissue, it's about microfragility. The physical gut lining is inherently weaker and tears more easily in people carrying this variant.
That makes so much sense. It's like building a brick wall, but the white pathway is telling the workers to use watered-down mortar. The wall might look totally fine on a sunny Tuesday, but the first mild gust of wind completely knocks it over. Did they find other genes messing with this bricklaying process?
They uncovered a whole suite of them. Genes responsible for how fast epithelial cells multiply or how they metabolize lipids. But the most surprising discovery in this one pathway was a gene called MYC.
Wait, MYC. Anyone who has even glanced at oncology research knows MYC. It is one of the most famous oncogenes out there. When MYC goes into overdrive, it causes cancer by telling cells to multiply uncontrollably.
Exactly. But here the script is completely flipped. In the context of a healthy gut barrier, you actually want cells to multiply to heal a wound. MYC is required for the intestinal stem cells to regenerate.
Oh, I see where this is going.
The study found that the IBD risk variant dysregulates MYC in these specific stem cells, completely preventing the gut barrier from healing itself when it gets damaged.
So instead of causing runaway growth like in cancer, the broken switch here prevents the necessary growth to heal an ulcer.
And it gets even crueler. They found another gene in this pathway, an interaction eQTL, called RNF14. The Crohn's risk allele drops the expression of this gene, which usually boosts the Wnt bricklaying process. But crucially, it only drops the expression when the gut is actively inflamed.
Oh, that is sinister. So right when the gut is actively burning from inflammation and desperately needs the bricklayers to work overtime to heal the bleeding tissue, this genetic variant pulls the fire alarm and sends all the repair workers home.
That is exactly what happens. The regenerative capacity of the tissue is literally destroyed by the very inflammation it is trying to survive.
That is a phenomenal insight. Okay, time for our final recap. Bullet 1. IBD is driven heavily by a failure to regenerate the gut barrier, not just by immune attacks. Bullet 2. The FIRM-T1 gene links incredibly rare tissue fragility disorders to common IBD. And bullet 3. Wnt pathway genes like MYC and RNF14 fail to repair the gut lining, shutting down repair mechanisms precisely when the body is under inflammatory stress. So what does this all mean for the patient listening to this right now? We finally have this 4K resolution map of the gut. We know where the Border Patrol goes rogue, and we know why the bricklayers go on strike. How does this actually change the medicine cabinet?
Well, first and foremost, it validates what we are currently doing in the clinic. The single-cell map confirmed that existing biologic drugs, like vedolizumab, which targets ITGA4, and afacitinib, which targets JAK2, work exactly on the cellular pathways we hope they did.
Which is always nice to confirm.
Right. But the absolute holy grail of a map like this is drug repurposing. Because they map the exact cell type expression, they found a specific gene, the PRKCB gene, that drives ulcerative colitis when it becomes overactive in monocytes.
And since you're bringing it up as a repurposing candidate, I'm going to assume there is already a drug out there sitting on a shelf that targets it.
Exactly. There are oncology drugs currently in clinical trials designed to specifically inhibit that exact gene. Because we now have definitive human genetic proof that this gene drives UC in these exact cells, pharmaceutical companies can potentially repurpose those cancer drugs for IBD, which saves decades of baseline research and gets treatments to patients years faster.
That is the dream scenario. But what about the opposite side of the coin? Like, one of the biggest complaints with any complex medication is the side effects. Can this 4K map explain why certain drugs meant for totally different parts of the body make our stomachs absolutely miserable?
Yes, the off-target side effects. If we connect this to the bigger picture, this was honestly one of the most elegant parts of the entire Nature paper. Take metformin, the incredibly common diabetes drug.
Sure.
It works wonders for blood sugar, but one of its most notorious side effects is severe diarrhea and abdominal discomfort. For a long time, we didn't fully understand why. The single-cell map shows that the ulcerative colitis risk allele drops the expression of a specific mitochondrial gene called NDUFAF1 exclusively in colonocytes. Well, metformin targets that exact same mitochondrial pathway. So when a diabetes patient takes metformin, the drug is inadvertently mimicking the exact cellular environment of ulcerative colitis in their gut.
My mind is blown. It's essentially inducing temporary, localized ulcerative colitis just by doing its job for diabetes. Are there other examples of this cross-wiring?
A much more severe one involves Alzheimer's disease research. A few years ago, a class of drugs called gamma secretase inhibitors were developed to treat Alzheimer's. But they failed dramatically in clinical trials
Why?
because they caused severe, unacceptable gastrointestinal bleeding in the patients. The single-cell map explains it perfectly. The specific gene that those Alzheimer's drugs target, called PSEN2, to clear plaque out of the brain, is also absolutely crucial for maintaining the gut epithelial barrier via, you guessed it, Notch signaling.
The walkie-talkies for the gut's border patrol.
Yes, by giving the patients a drug designed to fix the brain, they accidentally destroyed the Notch signaling in the gut epithelium. The border patrol lost communication, the barrier collapsed, and it caused severe bleeding.
That right there is the undeniable power of moving from the blurry bulk smoothie to the 2.2 million cell 4K resolution. It solves decades-old mysteries about diabetes and brain drugs just by looking at the neighborhood street addresses.
It really is a total game changer.
To wrap this deep dive up, the overarching lesson here is that diseases are not just a broken gene. They are a broken gene, hiding in a highly specific cell, reacting only under specific environmental conditions.
And that leaves us with a critical paradigm shift for the future of medicine. I mean, think about it. If a genetic variant only causes disease during active inflammation, and that inflammation dynamically alters how drugs interact with our cells, are we entering an era where your prescription changes not just based on your baseline DNA, but on the real-time, day-to-day inflammatory state of your specific tissues?
That is wild. For you listening, think about a medication side effect you've experienced in the past. Based on what you learned today about these cell-specific pathways and environmental triggers acting like chemical developers, do you think your genetics or your body's temporary environment was to blame? Let that marinate. The next time you see a medical scan or take a pill, remember you aren't just one static body. You are a stadium of trillions of cells, constantly reacting, each holding their own piece of the treasure map. Thanks for joining us on this deep dive. Keep questioning the world around you.